Doing these four today because these four are unique to plants only. You will not find these things in an animal cell. The central vacuole is used for storage, particularly retaining water. Water retention for plants is really important. It's what gives it some of its structure. When a plant wilts, it's because its central vacuole doesn't have any water in it because there's no water available to help the plant do its plant functions. So all of the water drained out of the central vacuole. When you water that plant and it unwilts, that's when the central vacuole is refilling with water. So it's when the whole plant cell refills with water. The presence of enough water inside of a plant cell to give it that structure, or to give it that firmness or rigidity, that pressure is called osmotic pressure or plasmolysis, or turgor pressure. So, turgor pressure is just the pressure that water inside a plant would exert on the cells to make them rigid and firm. Really super important in the life of a plant. Turgor pressure, if you don't replace the turgor pressure, so that wilted plant, if you go too long without unwilting it, it dies, right? It can't perform its life functions. So it's really important.
So like the turgor pressure is basically like giving it water?
Mm-hmm. It's the pressure that results on being full of water. So think about a water balloon. If you don't have any water in it, it's just like an empty balloon, right? If you put a little bit of water in it, it's not full all the way, and it's just kind of, like you're going to throw it at somebody, it's not going to explode, right? It's just sort of lame. The more water you get in it, the harder that balloon is, right? And that's when you whip it at somebody and it explodes, it really hurts because there's a lot of pressure inside that balloon, and when you release the pressure, it explodes. A plant cell is like that. When you put that much water in it, it gets very, very firm. That pressure is called turgor pressure. Plant cells have this awesome thing that protects them. Unlike the water balloon, which is more like an animal cell, a plant cell has a cell wall, so it's not going to explode. The only instance in which a plant cell is going to explode is if it freezes. The reason for that is ice crystals are pokey, right? You've seen snowflakes. Ice crystals are pokey, and it will stab holes in the cell wall. That's why frost kills plants, because it explodes their cells, because, of course, ice is larger volume than water.
What would happen when you overwater a plant?
A lot of times it won't absorb the water, so it just sits there on the surface and it'll cause rot. But sometimes if it does absorb too much water, inside a plant, inside that central vacuole, it relies on a very specific amount of certain minerals and vitamins, ions. Too much water makes too little ion, and then that kills the plant too. So that's why, you guys know much about plant care? There's like three essential things you can do in plant care. 1, underwater. 2, perfect amount of water. 3, overwater. It's like Goldilocks. You have 3 choices. Most people will commit the under or overwater problem. Very few people are perfect waterers. Both underwatering and overwatering are fatal problems for plants because it affects their cells. Their central vacuole goes empty. Or it's too full, and you end up with that problem of ions.
Shouldn't you water like the soil till it's, I think this word, moist?
It depends on the kind of plant. Every plant's different. It depends what environment the plant originally hails from. Chloroplasts, you've heard this one before, right? What happens in chloroplasts? Photosynthesis. The pigment chlorophyll gives it that nice green color that exists in chloroplasts. What you may not have heard before is that chloroplasts are actually made of very small discs that are all packed together. And those discs have funny names. So you ever see the word thylakoid or grana? They're referring to parts within the chloroplast. Thylakoid and grana are parts within the chloroplast. Inside of the chloroplast, chloroplasts aren't any particular fun shape. They're like, I'm trying to think of an analogous candy that is that shape. Almost like the Cadbury eggs. It is that time of year. They're like a Cadbury egg, and inside of them, though, they have little stacks, like of Smarties on top of each other, and those stacks are the grana. Chloroplasts also have some of their own DNA. Lends itself to the endosymbiont hypothesis that it must have been a free-living photosynthetic bacteria before it came to dwell inside of another cell. Plasmodesmata, tiny holes, itty-bitty holes that occur in the cell wall and cell membrane of two plant cells. They line up one cell to the other. They act as a passageway or method of communication between cells that are next to each other. And having those plasmodesmata, those pores, essentially, in the surface of the cell and cell wall, is important because we already said the cell wall, or with this being applied, the cell wall is there to keep plants rigid, right? To keep plants firm. And it's made of cellulose. So the cell wall in plants is made of cellulose. Remember we said that's what humans consider indigestible fiber when you eat plants. It provides structure. It is usually in like a nice geometric pattern that makes plant cells appear in very regular shapes. They're like small bricks or they're octagons. They're very, very geometric. Animal cells, not so geometric. They're kind of, well, depends on the tissue. Sometimes they're small and they look like Smarties. Sometimes they're long and they look like fibers. Sometimes they're tiny and they look like little trees. So animal cells, huge variety of shapes. Rarely are they geometric. Cilia, did we do cilia? Yep.
So you said the plasma,
Plasmodesmata?
or the pores inside of cells?
Yep. Last one I wanted to add. We did flagella, right? Or flagellum. Did we? Well, here we go. Flagella is, these two things occur on the outside of cells. That's why we saved them for very last.
Are these on both?
No. You are going to find flagella and cilia typically only on animals. Well, you'll find it on protists, bacteria, but you will not typically find them on plants. The reason for that is flagella and cilia are both methods of movement. A flagella is like a whip-like tail. In prokaryotes, they're very simple whips. In eukaryotes, they're usually more complex. But they, best way to describe it is like the propeller on an airplane. It spins in a circle and then pushes the cell in the opposite direction then. So just like on an airplane, it's going to spin and push. Cilia, on the other hand, are more like, say, here's my cell. Cilia are tiny little hairs that are on the outside of the cell. As opposed to a flagella, typically a cell is going to have one flagella, sometimes they're paired. Cilia, it's covered with thousands of them all around the outside of the cell. And they wave back and forth. And that's how they organize themselves in movement, is by waving back and forth. They also help certain cells sense things around them. They can sense movement of other things around them because if their cilia moves, they know there's movement around them. It's not much different than, what can I compare it to? the idea, if you have a breeze, you know the direction the breeze is coming from based on the direction that your hair moves, right? If you feel wind in your hair, you know which direction things are coming from and moving from because you can feel that movement in your hair. Same thing for the cell. It'll feel the movement in the fine hairs that coat the outside of it, and then it knows what direction things are moving.
I like the Fidelis, does it do anything special, or does it just push cells?
It just moves. So it's just I mean, it's an important function if you think about it, because cells do need to move from place to place to respond to their environment, but no special amazing function other than movement. Oh, let's see. Might as well do this one. Do it next chapter anyway. Inside of animal cells, there is this thing called a cytoskeleton. The cytoskeleton is exactly what it sounds like. So plant cells have the advantage of having a cell wall that provide it with shape. Animal cells don't have that, so it has to have something else to provide it with shape. Inside of an animal cell, it has all these organelles and it has cytoplasm, but it also has a miniature skeleton made out of microtubules and microfilaments. Microtubules and microfilaments. They're just different kinds of bones. But don't think of them as being very, very, very firm bones. Think of them more like the consistency of cartilage. If I were to take your forearm and attempt to bend your forearm, there's no bending there, right? That's too rigid. Don't think of something like that for a cytoskeleton. Instead, think of something like the cartilage in your nose. If you push it, it moves out of the way, right? But it still provides shape. It gives your nose a definite shape. The cytoskeleton of microtubules and microfilaments is firm and will give a cell shape, but also is compressible. And it allows multicellular organisms to exist. A lot of people don't think of this. When we look at something like that wall back there, it's bricks stacked upon brick, right? That bottom layer of bricks has to be able to support the weight of every other brick above it. And it's not as bad for the row above that because it only has to support one fewer layer of bricks, right? So the least amount of support are the bricks on the top. They don't have to be the most structurally sound bricks because they're not bearing any load. The ones on the bottom have to be perfect bricks because they're load-bearing bricks. Cells, if the ones in the bottom of that organism, the ones that support the weight from every other cell above it, if it didn't have a cytoskeleton, would get smashed because it's load-bearing. So it allows multicellular organisms to exist because it gives them the ability to bear a physical load. And when I say physical load, I mean like physics, like weight, structure. So it's kind of important. I don't know how accurate this is. I planned this, and we're still going to do it, regardless of what I find out right now. I'm assuming you guys have used microscopes before. Tomorrow, you're going to be looking at plant and animal cells under the microscope and comparing and contrasting. Friday, we review. Monday, we test. Microscope handling rules. They're really quite simple. Let's see what you can tell me about what you remember.
Carry it with 2 hands.
All right, 2 hands. Where are those hands?
One on like the handle part.
So one on the arm,
And the one on the bottom.
one on the base. So arm is this thing up here. Base is that thing down there. What else? Objective lenses.
Yes.
The objective lenses are the most expensive part of the microscope. A lot of microscope handling is intended on protecting the objective lens. We always store it on the lowest power. So the smallest one is how we store it. And we store it so the stage and the objective lens are as far apart as possible. We don't want anything to hit our lenses. When we focus, the only time we use the big knob, the coarse adjustment, is when we're on the smallest objective. Coarse adjustment moves the stage up and down. Can you guys see that stage moving? If we have high power objective and we move coarse adjustment, we get dangerously close to my objective hitting my stage. Very, very, very expensive damage if you do that. So coarse adjustment is for low power. Fine adjustment, baby knob, is for any other power. Any focusing rules you guys know? Now that we know when we can or cannot use coarse and fine adjustment. Any tips or pointers for how to focus? So when you take a slide and you scan it, do so on low power. So that means you move the slide back and forth looking for the object you're trying to magnify. Oh, a looking rule. A guaranteed way to leave here tomorrow with a headache and have one for the rest of the day. Look through the microscope like this. You have a muscle that surrounds your eye that forces it closed. That's what helps you close your eye, called the orbicularis oculi. And if you tense your orbicularis oculi for a long amount of time, it causes a tension headache in all the muscles that connect from the top of your skull down to the side. Really uncomfortable. And it's a tension headache, so it's not the kind of headache that Tylenol is going to make go away. So do me a favor, keep both eyes open. If you can't keep both eyes open and look through the microscope because you don't know how to make your brain focus out of only one eye, leave the second eye open, but cover it with your hand. As soon as light is blocked to that eye, your brain automatically switches. It's like, oh, I'm not using that eye anymore. So you don't have to think about it. Scan the slide on low. Center the object in your field of view. So center the object in your field of view. Here we're going to look at the letter P. You're just going to center it right there in the middle. That way when you switch objectives to the next highest objective, it's still in the center of your field of view. The center is what always gets magnified.
So I have a question about like the force and the,
Huh?
so if you wanna like change the magnification, do you have to put the force all the way to the bottom?
Nope. So say you found it and it's focused on low power. You're going to just look at the side of your microscope while you're switching. Turn it and just make sure that no contact is made. It must, whatever you see, must be in focus on low power in order to be able to have it focus on a higher power. If it's not in focus and low, never going to focus on high. High coarse adjustment, we already know store it, stage down, low objective, carry it with two hands, don't squint, eye patches, you're a pirate for the day, put your eye patch on. Is there any other way you could possibly break these? It's my job to consider these things. Yeah, don't drop them. Oh, arm of the microscope goes to your belly. Arm to the belly. That's how it goes on the tabletop in front of you. Not objectives to your belly. Arm to your belly. A lot of people use them backwards. Don't drag the microscope across your tabletop. Don't drag it across the tabletop for your partner to be able to look through it. What you'll want to do is make sure that you Move to the microscope, not the microscope to you. Typical rule that you have is don't use sunlight as your source of light. You can't, because these are the kinds of microscopes you plug in. Yeah. Does take a little more effort.
I'm not sure how
Oh, thank you. Oh, let's see. What else can I think of for microscope use? Don't drag it. When you plug them in, don't plug it in directly to the plug above your table. You're going to use a surge protector. So we're going to push our tables together in tables of 4 and have, actually you guys will be table of 8. And you're working in your partnered pair still, but you're going to have one surge protector for all of you to plug 4 microscopes into. So we keep our surge protectors in the cabinet that says Thurston Lava Tube. You can work alone, yeah, if you want. I warn you now, I will come over and help you find whatever object it is you want to be found under the microscope. I'm more than happy to do that. But do understand that practice makes perfect for microscopes. Don't give up right away. Yes, it's frustrating. Yes, it can be hard to find things, but it's easier in the long run if you learn how to do it now. I caution you of this because I'll come over and find the object right away. Been doing this for years. Had classes in college where for 4 hours at a time, all we did was look through a microscope. I better be good at it at this point. Me knowing how to do it is not going to help you learn how to do it. So I'm advising that you struggle a bit before you ask for help. Not because I enjoy watching you struggle, but because I know that it's a learning thing. To determine magnification that you're looking at, eyepiece times objective lens. So here my eyepiece has a magnification of 10 times, and my smallest objective lens is a 4-time magnification. So on low objective, it's magnified 40 times, 10 times 4. Medium, and by medium on mine, I mean medium 1, because I have 4 objectives. You guys in your microscopes will only have 3. We have 10, so 10 times 10, 100. 10 times 40, 400. And then high power, we have 10 times 100.
It's gonna be 1,000.
1,000 time magnification. What else do you need to know? Oh. As you increase objective, your field of view gets smaller because you're zooming in on the center of the slide again. So your field of view will get smaller and darker, technically. So I am not having you guys do an intro to microscopes like, look at this piece of newspaper under the microscope, because I'm assuming you've used microscopes before. Yeah? I don't want to insult your intelligence. Like, I'd rather just give you living cells and be like, here is the living cell. Look at it. Have you prepared wet mount slides before?
Yes.
Okay.
Yes.
Quick primer. You'll get a blank slide. You're going to take the cell, whatever it is I ask you to look at, you're going to take it, put it on the slide. In some instances, that means you're going to swab the inside of your cheek, swab it on the slide, put a drop of water on it, and then lower the cover slip on at a 45-degree angle and lower it down. That prevents air bubbles from getting trapped between the slide and the cover slip. Same thing for your plant cell. You'll do the same thing. Then you're going to have to stain them. The animal cells you're looking at tomorrow, you have to stain with a lovely blue stain. Do me a favor, it's intended to stain animal cells. You are an animal. So if you get it on your hands, it will stain you in a lovely shade of Smurf. You will look like you went on a Smurf smashing rampage if you get it on your hands. So do me a favor, try not to get it on your hands. It will come off in a few washings, so like a few days. So
Is it bad for you?
it's not bad for you. I mean, don't drink it, but it's not gonna poison you through your skin though. It just, you'll look like that kid who got the methylene blue all over your hands in biology for a few days.
That'd be fun.
So, you know, and the animal cell or the plant cell you're going to dye with a substance called Lugol's iodine. Lugol's iodine won't react with your skin too badly, so you don't have to worry about that one so much. It will react with your clothing though, so try and not get it in your uniform polos. The dyeing technique is a little more complicated. I'll demonstrate it for you tomorrow, because it is a little more complicated. So, there's that. You have all of the information, data, etc. now that you need for your test on Monday. My advice for you, homework, is that sheet that I just gave you. We are in lab tomorrow. We review on Friday. We test on Monday. Make sure you go back through the video that's on Plus Portals for the purposes of studying. Make sure that all of your notes contain all of the information in that video. Just to make sure that we supplement between the two. There will be some microscope handling stuff on it because, well, quite frankly, this should be the easy points, right? I mean, you already told me how to handle a microscope. Probably not a microscope labeling thing. I'd rather you know how they work rather than what they're called. So, you and your partner may